All right, let's continue on the chapter 26. This is part 2. So we take the geologic record, the historical Earth, if you like, and it's separated into 3 eons: the Archean, the Proterozoic, and the Phanerozoic. Those are probably less important than some of the other things we'll talk about in a second. There's lots of eras and lots of periods that are that those are subdivided into, and most of them are marked by major changes with fossils. So here's the overall geologic record, and on the left you can see the eons that we just talked about, the major subdivisions, and we'll talk about a few of the more specific important aspects of that. So using the fossil record, we've been able to identify a number of drastic environmental changes that caused a major shift in species number or in species type, whether it be extinction or large growth or accumulation of species. And those are actually marked here, and as we talk about them. There's the Permian extinction, okay, which marks the transition from the Permian to the Triassic periods, and then we have the Cretaceous mass extinction between the Cretaceous and the Paleogene. Again, the second important one. So those are the 2 mass extinctions. The Permian mass extinction is well known because about 96% of all marine animal species were killed off, and also 8 out of 27 orders of insects. So tons and tons of animals were killed, and the cause is believed to be due to volcanic eruptions. The other one is the Cretaceous time period. This is the dinosaur extinction. The other one is smaller animals, non-dinosaurs. So here we got a large number of deaths as well, marine and terrestrial, and again, dinosaurs is the most memorable one. And this one's thought to be caused by meteor impact, causing release of debris into the atmosphere that covered the globe and thus affected photosynthesis and the food chain. We still don't know enough. But we do know that there's every time some of these drastic events occurs, that it promotes radiation and specifically speciation to occur because some are better fit and able to survive, and they can fill those newly vacated ecological niches because there's no animals occupying it, no species. Some people like to view the geologic record as a time clock, defined by the major events again. And so here we have the origin of the Earth and solar system, and then going around clockwise, we start the first living thing with prokaryotes. Prokaryotes are the cause of the release of oxygen into the atmosphere, which enables single-celled eukaryotes to survive now. These prokaryotes are not bacteria, but archaebacteria. That are able to survive in harsher environments because the environment had no oxygen or little to no oxygen until this point. Then we get single-celled eukaryotes followed by multicellular eukaryotes, and then we get what we commonly think of animals and land plants, and then finally that little teeny tiny sliver is us, is humans. So that makes us feel very significant. 26.3, so into prokaryotes. The oldest known fossils are called stromatolites. And they actually look like rock, but they are bacteria that are ingrained into part of the sediment. And the oldest one that we've dated is 3.5 billion years ago. And so here's a few pictures of stromatolites. The book, if you look at these pictures in the book, it's a little bit more, a little bit easier to see. But this is in Baja, and this is in Australia, dating stromatolites. Prokaryotes, as you saw on the clock, were the Earth's sole inhabitants for about 1.5 billion years, quite significant. And they actually developed our first electron transport systems, and they still occur in those bacteria today, although they're far more simplified, and they don't occur on any chloroplast or mitochondrial membrane, of course. They occur in prokaryotes on the cell membrane. They're essential to life, of course, and they're tied to photosynthesis. Photosynthesis, as we learn in Chapter 10 in a week or two, there is an electron transport chain on the membrane of the chloroplast, which mimics those of prokaryotes, or is very similar. The earliest types did not produce oxygen, and thus the reason why those early prokaryotes did not release oxygen into the environment. 3.5 billion years ago, we had our initial production of oxygen, and this was from cyanobacteria that actually did oxygenic photosynthesis, and you can see some of those remnants here with iron deposits and so on, the various colors. And that picture is again pretty easy to see in the book. So oxygen began to accumulate in the atmosphere, and it was actually really challenging for life because most things had never experienced oxygen, and oxygen can oxidize substances. But this enabled them now to be able to gain energy from light photosynthesis, if you like, and we could now enter new ecosystems. And here now we have the rise of eukaryotic cells due to the endosymbiotic theory, we believe. That's the hypothesis, at least, where there's that communication and symbiotic relationship between prokaryotes. And here you see how complex eukaryotic cells evolved from much simpler prokaryotic cells. The oldest eukaryotes are 2.1 billion years ago, and here's the theory of endosymbiosis, where mitochondria and plastids are the large family of things which includes chloroplasts. They were formerly small prokaryotes living inside larger host cells, and thus a symbiotic relationship inside, endo meaning inside. So they probably gained entry to the host basically as parasites, or they were drawn in through endocytosis but not digested enzymatically, and then survived and lived on inside, as you can see here. Here we have the nucleus forming within, also potentially due to endosymbiosis, although that's another topic, another issue. And here you see mitochondria. This is a prokaryote, actually, which will become the mitochondrion, and then they engulf photosynthetic prokaryotes like cyanobacteria to become these plastids or chloroplasts. And you do notice that nucleus came first, followed by mitochondria, followed by chloroplasts. So there's a significant amount of evidence. There's a long, long list. This is just a small, shortened portion of it. The major aspects being that there's an inner membrane for both mitochondria and plastids. They have a double membrane, inner and outer, and both are important for their processes energetically, which we'll talk about at a later date. And as a separate note, they also both have their own circular DNA, and even on top of that, they also have their own plasmids, much like prokaryotes do. So you know that mammals have linear DNA in the form of chromosomes. Well, prokaryotes have one circular chromosome, and so do mitochondria and plastids, and they're about the same size. We do also know that prokaryotes have the ability to transfer genes from one another or to one another. We'll talk about that again at a later date, but it includes conjugation and transduction and various processes, and this probably contributed to the ability to increase the size of the genomes, because a eukaryotic genome is much larger and more complex than a prokaryotic one, and thus enabling eukaryotes to be more complex. So this ability to transfer genes from one to the other potentially enabled this to happen. Some investigators think that eukaryotic flagella and cilia, those structures sticking out of this cell membrane here, actually evolved from symbiotic bacteria as well, between bacteria and protozoans, and thus enabled locomotion to occur. And we'll take a pause on this one.